Hello.
Yes,
So,
yes,
yes,
sir.
sir.
Yes,
Yes,
sir.
sir.
Yeah.
Yes, sir.
Yeah.
Yes, sir. Audible.
So I told I'll be speaking on some of the outlooks and challenges in life sciences in this particular inaugural address. Now, until 2013, the biological specimens were identified by using morphological features like shape, size, color of the body parts. Like in plants, the different characters, morphological characters were used, whereas wherein a trained taxonomist was required to identify this. In the year 2003, Paul Herbert, a researcher from Canada, he proposed DNA barcoding to identify a species. And DNA barcoding is a very powerful tool to assist scientists to catalog the world's biodiversity. It uses concise genetic sequence from a standard genome part. It is a powerful tool in helping scientists to catalog, as I said, the world's biodiversity. Even non-specialists, other than taxonomists, can obtain barcodes from very tiny amounts of tissue using DNA barcoding. It is a new tool in the taxonomist toolbox, helping them to quickly identify plants, animals, and microbes. So here the challenge would be to conserve the hotspots of biodiversity and the fragile habitats, especially in the era of global warming. We know that the temperature of this planet is slowly increasing, and we have a lot of problems in the present times. So, and then we have no count on the biodiversity that is existing. And even before knowing the biodiversity, if it is lost, then then it's a problem. Again, coming to the Nagoya Protocol on assessment and benefit sharing that was adopted in 2010 and it came into force in 2014. Now, this is regarding, the objective of this is fair and equitable sharing of benefits using genetic resources. So, thereby helping to conserve towards conservation and sustainable use of biodiversity and human well being. So, this it covers the Nagoya Protocol, it covers the traditional knowledge associated with genetic resources. Now, the genetic resources are already available. The traditional knowledge is with the locals, is with the people, and therefore they have to be benefited. So, whatever the traditional knowledge that is existing in this population with this people has to be shared, and then benefit should go to the locals. Then we know about the biopirates. Biopirates, they benefited and prospered by plundering the natural resources of the developing and less developed countries without paying any royalty to the source countries. Recently, there have been several cases of biopiracy of traditional knowledge in our country. Some of the classical examples include the turmeric, basmati, then neem, and brinjal. According to Stephen Willey, a system biologist at Pacific Northwest National Laboratory, two areas are poised for explosive growth. And these two areas includes one is the single cell biology and the other is CRISPR technology. Now, single cell biology deals with single cell sequencing, single cell proteomics, and single cell imaging. They are a part of this single cell biology, which will definitely make an impact. It is a fact that cells in the population are very heterogeneous, and what we see in the at the population level is the cumulative reflection of what the individual cells are doing. We can make a difference only when we understand what the individual cells in the population are doing. For example, uh, cancer heterogeneity. Treating a cancer cell, cancer disease, the ailment, it can kill 95% of the malignant cells, whereas 5% will still remain. And as a result, there is recurrence of cancer. So, unless one really understands why these resistant cells are different, one can never develop an utterly effective treatment against cancer. With that, technology in place, both sequencing technology and mass spectrometry technology, we only need to look at individual cells. Coming to the technology, CRISPR stands for clustered, regularly interspaced short palindromic repeats. Researchers regularly use CRISPR in the laboratory to alter genes in plants, bacteria, as well as in animal models. One can directly observe the affected traits if one knocks out a particular gene in lab mice. So, this technology could hold a great potential as a treatment for human diseases. In theory, CRISPR could let us edit any gene mutation that will occur in any disease with a genetic origin. So, therefore, this particular technology, is very important. So, both these technologies that I've discussed, they are revolutionary. Now, coming to the higher education has become essential for our prosperity and quality of life in the knowledge based age. According to NEP 2020, we must develop skill based components in the syllabus. So, with new industrial revolution, that is Industry 4.0, uh, that has begun in the year 2010, it includes new technologies like uh, the AI, then IoT, Internet of Things, drones, 3D printing, robotics, among several others. So there is a need to integrate these technologies into the various disciplines so that the students are abreast of these developments and they are equipped. For this, the syllabus and the curriculum should be updated, wherein we need to include these technological advances. Gone are the days where the industries envied one another. So they used to envy one another for profits. So in the present time, industries practice something called as collaborative competition that would create a win-win situation for both the industries. So that is what the industries are looking at. Well, I'm delighted to record the services rendered by the UGC HRDC Center of Goa University. It has undertaken this fascinating and commendable service to improve the academic standards and career development of the teaching fraternity, which would transform the students. Our HRDC is continuously engaged in organizing the UGC sponsored courses throughout the year. We sincerely thank the UGC for their continued support. So let enriching of teachers and the resultant empowerment of students be the motto of this course. Also, as teachers, we need to play a very significant role in the environmental protection by bringing environmental awareness among the students, community, and society. So, students are important. We need to train them so that they can participate in the environmental protection. I welcome all the participants who have come across, from the different states of our country, and I hope that you will have fruitful deliberations during the course. I wish you all the best, and I thank you for your patience. Thank you.
Thank you so much, Professor Bernard. On behalf of the RC in the Life Science, I'd like to extend a hearty vote of thanks to Professor Bernard, who spared time from his busy schedule to grace this occasion. We had the opportunity to really hear your thoughts, and this will encourage our students. Professor Bernard spoke on biodiversity, CRISPR, cancer cell, advances in higher education, updating curriculum. Thank you so much, sir, for sparing time to encourage our participants. We
Thank
would
you.
also like to thank our Vice Chancellor, Professor Harilal Menon, for allowing us to conduct this refresher course in an online mode. Professor Purva Hegde Desai, our in charge director, Professor Garg, our course coordinator, Professor Bernard, our chief guest, the HRDC office, and all our participants who have joined for this course. We request you to please turn on your cameras so we can take a screenshot of this memorable event.
Thank you, Professor Bernard, for accepting the request and
Thanks, Professor Shinde.
present for the session.
Thank you.
Requesting all the participants to please turn on your cameras. Each participant can also take a screenshot for themselves by themselves. Thank you, everyone. Thank you, Professor Banan.
Yeah, thank you.
The inaugural function has come to an end. We may now proceed with other instructions. Professor Garg?
I request to all the participants be present for session before time, and if there is any problem, please contact the coordinators or the HRDC directly on the phone, so that that can be resolved, issue can be resolved immediately. And always please take notes when the session is there, because as Ingrid had said, that you will not receive any material from our side. So then it will be difficult for you. If you miss the session, then it will be difficult for you to answer the MCQs, because it will be from all the sessions. And for presentation, we will send you the schedule, Ingrid, by today itself.
Presentation session? Yes, we can do that.
So that they are ready, because I have to share with those resource person also
Yes.
for the evaluation.